Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this session to discuss NCERT Class 12, the chapter Molecular Basis of Inheritance, Part 15. The fragments were sequenced using the automated DNA sequence that is worked on the principles of the method developed by their Frederica Sanger. So, in this total principle method is developed by scientist was whom? Frederick Sanger. Frederick Sanger. And here, remember the singer is also is credited at the credit completely created for as in developing methods for as in determination of the amino acid sequence of any proteins. So how do the amino acids as present? How do their sequence is present? Everything their the Frederick Singer he noticed. Okay, these sequences where they are completely arranged based on the in some their overlapping region present in them. So this completely that enter the genetic information they are overlapping to as in they are codons. Okay, like next this required a generation of overlapping fragment the sequencing. So here the how do they overlapping how they complete the overlapping line the overlapping line. Okay, next element of this sequence was humanly not possible. So in this sequence we are not doing properly here, therefore they specialized computer based program where they develop. Okay, so this total we get using as in the computerized, we are using as a computer based program we are developing these sequence where they're completely subsequently they are annotated and where they're assigned to of each chromosome. So each chromosome they're completely they get assigned which chromosome, say here which base pairs to get attached to as in which regions they get completely computer automatically they're generated. The sequence of the chromosome it first the chromosome 1 was there completed only in the May 2006. It got lost of the in 20 for human chromosomes, that is in 22 autosomes, and that is in X and Y to be they are sequenced. So the X chromosome and Y chromosome they are completely their sequence. Another challenging task, the completely task was assigning as in the genetic and the physical mapping on the genome. So how to the physical mapping? How to there we get developed as in the physical mapping? They are physically they are challenged. It was generated as using the information, the polymerization of the restriction endonuclease, their recognition sites, and the some the completely the repetitive DNA sequence known as in that microsatellite, which is the applications of any the polymorphism in the DNA receptive of the DNA sequence shall be there explained into the next section of the DNA fingerprinting. So in this, how to the where the DNA polymerase enzymes get participated in which where the DNA enter the sequencing will that takes place, where is present as in that in complete microsatellites, how do they get working, their entire we discussing that in a completely next to the DNA fingerprinting. Okay, this is silent features of human genome. Silent features of human genome. In some the silent observations draw from the human genome project or as follows. So some that human genome project they are following as in some silent features. Once we see the one by one. The first one, the Human Genome Project contained in a 3164.7 billion base pairs. So their human genome contain how many? 3164.7 billion base pairs. The second one, the average gene consists of as in 3000 base pairs, but the size is very greatly with the larger, the largest known as a human gene. They begin their dystrophin at 2.4 million base pairs. So here the completely their dystrophin it has in how much 2.4 million base pairs. Next, the total number of genes is in estimated as in a 30,000, much lower than previous estimates 80,000 to 1,40,000 genes. So here the ones we observed as an estimated and 30,000, but the previous estimated how much 80,000 to as in a 1,40,000 genes. almost as in a 99.9% the nucleotides base space is completely exactly has in a the same in all people. So all people almost they contain as in a this kind of the million base pairs as present. Okay, next, the function are unknown the over 50% of any discovery genes. So around as in 50% of genes were discovered. Less than 2% of the genome code for the proteins. So mostly the 2% the genes they code for as what? Protein. Code for protein. Next, here the repeated the sequence make up very large portion that is human genome. So the number of here the sequence will there repeatedly they are complete large portions is present in the human genome. Next, the repetitive the sequence of the DNA the stretches of the DNA sequence that RNA the repeated many times. So the number of many times the DNA will be they get repeated, and the sometimes 100 to 1000 times they are in a through the have the no direct coding functions, but there shall be the light on the chromosome structure and dynamic and evolution. So mostly the DNA, for example, when you take a some particular DNA, this DNA the number of time they get repeated. Okay, the each and every the chromosome structure automatically will be get changed. Means the chromosome structure will be they get the base pairs are to completely get replace the number of enter the DNA, because here what happened the structure and dynamic and the evolution will be they get changed depending upon the sequence. The chromosome 1 has in the most genes around as in 2968, and Y chromosome has in the fewest 231 only. Okay, here X chromosome, Y chromosome is in a so like. Next, the scientist has in the identified as in 1.4 billions located as in that single base DNA differences, that is in a SNPS, means that is same the single nucleotide polymorphism, that is in a single nucleotide polymorphism. Next, it is pronounced was in the synapse. Next, it occurs as in only for humans only. Next, this information the promises to complete revolutionist process that finding the chromosomal and located for as in the diseases associated with sequences and the tracing the human history. So these are mostly which purpose they helpful. It is mostly helpful as in to tracing as in that human history. Human history. Okay. Next, what are the applications and the future challenges? What is the applications and future challenges about as in the human genome project? Let's see here. The driving that meaningful knowledge from the DNA sequence to define the research through the in a coming their decades, leading to our in understanding of biological system. So it is when you go to as a research field, what happened the DNA sequence it is complete they get difficult. Next, these enormous talks will be there require the completely expressed to as in the creativity that tens thousands of the scientists from as in a varied their disciplines in that both and public and private sectors in worldwide. So the enormous talks takes place in creatively in 10,000 of in a scientist validate the complete varied their disciplines in both public and completely private sectors in the total worldwide. Next, one of the great the impact of having as in a human genome sequence may be there, enabling completely directly is a new approach the biological research. So in the biological research, they complete their approach one by one. New things will be their approach, and in the past, the researchers studied one of the few genes at a time. So one at a time, there are number of genes to get noticed. What happened? It is take make as a confusion also. Okay, with his old genome sequence and the new high the throughput technologies can be the approach the question that systematically and on a much their broader scales. So the number of their development was takes place. What happened? It is takes place as in that difficulties. They can study all genes in genome. For example, all in transcripts and particular tissue or organism or tumor, or how the 10,000s of genomes the project the protein work together interconnected network to completely architect their chemistry of life. So here the number of their tissues, so organisms, so tumors, so cells, then at a time they get participate, what happen? It is to completely, it is attached to as in a chemistry life. It is a complete network like structure. We discuss in next class. Thank you for watching. Please share and subscribe.